everyone, I'm Swati Shrestha, a second year PhD student with Dr. Jeffrey Meru in Vegetable Breeding and Genetics Lab at the University of Florida. My PhD research focuses on development of aphid transmitted virus resistant squash and pumpkin cultivars using molecular breeding approaches. Aphid transmitted viruses like zucchini yellow mosaic virus and papaya ring spot virus are the major yield limiting factor in squash production worldwide. Development of virus-resistant cultivars using traditional breeding relies on rigorous and resource-intensive phenotypic assays. To overcome this problem, I'm working on identification of quantitative trait loci and molecular markers associated with virus resistance that would assist in phenotypic selection, thus accelerating the disease resistance breeding.
Hello everyone, I'm Swati Shrestha, currently a PhD student with the University of Florida, and today I'm going to talk about my research titled QTL Identification and Marker Development for Papaya Ring Spot Virus Resistance in Squash. To begin with, a little background on squash and pumpkin. Squash and pumpkin belong to family Cucurbitaceae and genus Cucurbita. And as you can see in the pictures here, these are diverse phenotype with versatile use and nutritional benefits. Potyviruses, which are the aphid-transmitted viruses, are the major yield-limiting factor in squash production worldwide, and with climate change, these viruses are getting more severe. Major potyviruses infecting squash are zucchini yellow mosaic virus or ZYMV, papaya ring spot virus or PRSV, and watermelon mosaic virus or WMV. And out of these three viruses, today I'll be talking about papaya ring spot virus or PRSV. Now, the symptoms of papaya ring spot virus include severe mottling of the leaf and narrowing of the leaves, and these can also infect the fruits, leading to deformed fruits, as can be seen here. Since viruses are obligate intracellular parasite, the best option for controlling the virus is genetic control or use of virus-resistant line. Now, virus-resistant line can either be developed by using traditional breeding approaches where the phenotypic assays are tedious and time-consuming, or they can be developed by using molecular breeding approaches where the selection of disease-resistant individuals at each generation can be done with the help of molecular marker, which is easier and faster. However, the markers significantly associated with PRSV resistance in squash are currently unknown. So based on this background information, the objective of the study was to identify QTL associated with PRSV resistance in squash using whole genome resequencing based bulk segregant QTL-seq method and develop molecular markers associated with PRSV resistance. The objectives were based on the hypothesis that advancement in next generation resequencing techniques will enable rapid discovery of genomic regions associated with PRSV resistance in squash. Moving towards the materials and methods, and the first step in the study was development of mapping population. For this, PRSV resistant wild line from Nigeria called Nigerian local was crossed with PRSV susceptible commercial line called butterbush. The F1 were then self to produce the F2, and these F2 were mechanically inoculated with the PRSV virus. The response of the F2 individuals to the viruses was done on a scale of 0 to 3, where 0 meant no symptoms and 3 meant most severe symptoms. On the basis of the response of F2 individuals to the viruses, 10 most resistant individuals were selected and called as resistant bulk, and 10 most susceptible individuals were selected and called as the susceptible bulk. The disease confirmation in these bulks were done by using the ELISA. After selection of the resistant and susceptible bulk, the next step was whole genome resequencing and QTL analysis. For whole genome resequencing, DNA was extracted from parents and resistant and susceptible bulk and sent for sequencing. After getting the sequencing results, a series of bioinformatic analysis was done to identify the SNPs associated between the resistant and susceptible bulk. And after identification of SNP between the resistant and susceptible bulk, QTL analysis was done by using QTL-seq-R software. After QTL identification, the markers associated with resistance were developed by using Primer 3 Plus software. Now moving towards the results and discussion and starting with the phenotyping of the mapping population. A total of 118 F2 individuals were evaluated for the response to virus. And as you can see in the figure here, the individuals with the disease rating of 0 were perfectly healthy without any viral symptoms 28 days after inoculation of the virus. However, the individuals with the disease rating of 3 showed distinct viral symptoms and stunted growth 28 days after inoculation of the virus, as can be seen in the figure. Out of the total 118 F2 individuals, 54% were susceptible and 46% were resistant. And the chi-square test revealed that the ratio of susceptible to resistant was 9:7. And the similar results were obtained from previous inheritance study in squash in 2019. Now moving towards the results of the QTL analysis, and here we can see that a single QTL associated with PRSV resistance in squash was identified in chromosome 9 at 95% confidence interval. This green line here is the 95% confidence interval. A little more zoomed in version of what's happening in this chromosome 9. So this graph here is the genomic region of the susceptible bulk. This graph here is the genomic region of the resistant bulk. And this graph here is the difference in the genomic region of the resistant and susceptible bulk. The region here inside the red bars shows that this region is significantly different between the resistant and susceptible bulk at 95% confidence interval, which means that this region here inside red bar in chromosome 9 is associated with PRSV resistance. So after identification of this region, we developed CASP marker from this region. And out of these CASP marker developed, 4 CASP marker here highlighted in red were found to be significantly associated with PRSV resistance in F2 population. So here's the figure of the amplification of the CASP marker 1 and we can see here that they were able to beautifully distinguish the resistant and susceptible individuals in our F2 population. So the conclusion and future implication of the study. Now, from the study, we were able to conclude that QTL-seq method was efficient in identifying QTLs associated with PRSV resistance. We were able to develop 4 CASP markers significantly associated with PRSV resistance from the study, and then these markers will be routinely used for selection of PRSV resistant individuals in our squash breeding program. With this, I would like to thank you all for the attention, and I am ready to take any questions. Thank you.